Welcome to GenoTech, where science meets simplicity and sometimes a little humor. Today we're diving into the fascinating detective story of microRNAs and how researchers figure out whether these tiny molecules are secretly regulating our genes. Think of this as a crime investigation show, but instead of detectives chasing criminals, we have scientists chasing short pieces of RNA, just 22 nucleotides long, lurking inside the 3' UTR of messenger RNAs. Yes, the 3' UTR, the quiet end of a gene's message that doesn't code for proteins, but holds all the juicy secrets of regulation. If DNA is the blueprint and proteins are the workers, then the 3' UTR is like the HR department. It decides who works, when they work, and how much work gets done. Let's start with the basics. MicroRNAs or miRNAs are tiny non-coding RNAs, about 22 nucleotides in length. They don't make proteins themselves, but they love to boss around the proteins that do. Imagine a kitchen. The chef is mRNA, busy cooking up a protein dish. miRNAs. They don't cook. They just lean over the counter, arms crossed, and say, maybe don't cook so much. In fact, slow it down. That's exactly what they do. They bind to complementary sequences in the 3' UTR of messenger RNAs, and once attached, they can either block translation or destabilize the mRNA, making sure the protein output drops. So, tiny RNAs, big drama, they are the regulators behind the scenes, fine-tuning our genome. Now, why the 3' UTR? Think of the 3' UTR like a playground at the end of a gene. It doesn't directly make proteins, but it's full of swings, slides, and climbing frames for regulatory elements. Here, microRNAs recognize their favorite target sites. And because the 3' UTR is non-coding, nature has used it as a billboard for signals: stability, localization, and regulation. To complicate things, some genes even have multiple 3' UTR isoforms. Long ones with plenty of miRNA sites and shorter ones that dodge regulation altogether. It's like Netflix subscription tiers: basic plan with no ads, or premium plan with all the control. So, how do researchers find out if a microRNA might be targeting a gene? We don't start in the wet lab. That would be expensive, slow, and sometimes as frustrating as waiting for a Windows update. Instead, we start with computational prediction, and here's where the fun begins. Algorithms. Each tool is like a detective with a different personality. Target scan, the seed match detective. It looks for perfect matches in positions 2 to 8 of the microRNA. Very picky, very precise. Bonus points if the site is conserved across species. miRNA, the energy miser. It aligns microRNAs to the UTR and calculates binding energy. Lower free energy, stronger binding. Basically, it's always looking for the cheapest deal. RNA hybrid. The thermodynamics geek. It doesn't just check if they match. It asks, how stable is this pairing really? PITA. The accessibility police. Even if a sequence looks perfect, it won't matter if the RNA is folded up tight. PITA says, if you can't get to it, you can't use it. miRMAP. The all-rounder coach. It integrates multiple features: conservation, energy, accessibility, and gives you an overall score. Now here's the rule of thumb. Never trust just one detective. Using a single tool is like checking your horoscope in only one newspaper. The prediction might be fun, but it's not reliable. Instead, researchers cross-reference multiple tools and look for consensus. If 3 or 4 different algorithms point to the same site, now we're talking about something worth testing. Predictions are good, but evidence is better. This is where databases come in, armed with experimental results from previous studies. Starbase, full of ClipSec data showing where Argonaut proteins have been physically bound. PostR, rich in post-transcriptional regulatory data. miRTARbase, a goldmine of experimentally validated miRNA target interactions. Here's the analogy. If computational tools are your Google Maps directions, these databases are like actual traveler reviews. Yes, I've been there. Yes, the road is real. And yes, the restaurant at the end does serve great food. When predictions overlap with experimental evidence, confidence shoots up. But hold on, computational predictions in databases can only take us so far. At the end of the day, science demands proof in the lab. The gold standard? The dual luciferase reporter assay. Here's how it works. Take the 3' UTR sequence of your gene of interest. Clone it downstream of a luciferase reporter gene. Then introduce this construct into cells along with your candidate microRNA. If the microRNA really targets the sequence, the luciferase light dims. Create a mutant version of the UTR, one that disrupts the seed match, and the light stays bright. Boom. Specificity confirmed. It's like a light switch test. Add the microRNA, lights go out. Remove it, lights stay on. Other methods include measuring microRNA and protein levels after adding miRNA mimics or inhibitors, immunoprecipitating Argonaute complexes, and checking if your transcript shows up in the mix. These experiments are like catching the microRNA red-handed at the... Of course, biology loves to complicate everything. Multiple miRNAs on one transcript. They team up like a group project, piling onto the same mRNA to amplify the effect. Alternative 3' UTRs. Some transcripts escape regulation by using shorter isoforms that hide the target sites. Sneaky. Isomers. Slightly modified versions of microRNAs that shift the seed region, changing the rules of engagement. A single typo, and the whole password changes. So detecting microRNA interactions isn't just about finding a match, it's about understanding the full context. Let's recap the detective work. Start with your gene sequence. Run multiple computational prediction tools. Cross-check predictions with experimental databases. Pick the strongest candidates. Validate them in the lab with luciferase reporters or Argonaute IP. Consider biological complexities like isoforms and cooperative effects. It's simple, right? Well, simple in the way juggling flaming swords is simple. So there you have it, the full journey of detecting microRNAs in the 3' UTR of genes. It's part computational detective work, part experimental proof, and part wrestling with biology's endless surprises. Thanks for joining us today at GenoTech. If you enjoyed this mix of science and storytelling, don't forget to like, share, and subscribe. Stay curious and remember, even the tiniest molecules can make the biggest difference.